and I'm representing the Office of Bioequivalence in the Office of Generic Drugs. And today I'm going to be talking about best practices for conducting bioequivalence studies. And so before we get into the details of the presentation, I'd like to talk a little bit about some of the topics that we'll be covering today. First, I'd like to discuss the definition and the importance of bioequivalence in the world of generic drugs. And then transition into talking about some of the regulations that define bioequivalence and give the agency the authority to ask for bioequivalence studies. Then speaking of bioequivalence studies, we'll then shift over towards talking about the different types of BE studies that may be submitted in support of abbreviated new drug applications, or ANDAs. And then once we describe those studies, we'll talk about some of the tools that the agency provides to help industry in designing the appropriate approach to demonstrate bioequivalence. And one of the main tools that we provide are the product-specific guidances, or I'll refer to them as PSGs for short at points during the talk. As many of you may know, in vivo or in vitro BE studies are a big part of demonstrating bioequivalence, but in some cases, we might also entertain a waiver of the demonstration of in vivo bioequivalence. So I'll walk through some of the different waivers that we might consider. Then I'll take some time and focus a bit on the biopharmaceutics classification system-based waivers. And then finally, towards the end, we'll explore some of the different aspects of your submissions that we hope will help both the agency and industry submit complete, high-quality bioequivalence submissions. So first, let's start off with just laying the foundation, bioequivalence in generic drugs. So in the United States, generic drug must be pharmaceutically equivalent and bioequivalent, and thus therapeutically equivalent to the reference listed drug product in order to be approved. And this concept is laid out in the Code of Federal Regulations, or I may refer to it as the CFR for short. So as I mentioned, we have these two pillars. We have pharmaceutical equivalence, we have bioequivalence. So pharmaceutical equivalence are drug products in identical dosage forms and routes of administration that contain identical amounts of the identical active drug ingredient. And bioequivalence, which is the focus of this talk, is defined as the absence of a significant difference in the rate and extent to which the active ingredient or active moiety in pharmaceutical equivalents or pharmaceutical alternatives becomes available at the site of drug action when administered at the same molar dose under similar conditions in an appropriately designed study. And again, this is taken from the CFR, the Code of Federal Regulations. And so then, again, the concept is if pharmaceutical equivalence and bioequivalence is established, then you've demonstrated therapeutic equivalence. As I said, there are a number of different regulations that govern bioequivalence studies. And I'm not going to go through all of them, but here's just a highlight of a few of them. And they, first of all, in some cases, the regulations actually give the agency the authority to ask for the bioequivalence studies. And there are other regulations that describe bioequivalence and bioequivalence studies and so forth. So of this list here that you see on this slide, I think really the critical one is the last one, and that's 21 CFR 320.24a. And first of all, it gives the first part of it. It says FDA may acquire in vivo or in vitro testing or both to measure the viability of the drug product or establish the bioequivalence of specific drug products. I think the key point here is this underlined point, which says applicants shall conduct bioavailability and bioequivalence testing using the most accurate, sensitive, and reproducible approach available. And this is a really important point because this should inform the type and design of the study used to demonstrate bioequivalence for a given product. So in this regard, there are a number of different types of BE studies that are out there. But as I'll sort of emphasize in the coming slides, it's not one size fits all. Different products are going to be, or different types of studies are more relevant for certain products than others. So the general approaches are outlined in 3.20.24b, and I've sort of summarized them here. They can range from pharmacokinetic studies, where you're measuring the active release from the formulation in the systemic circulation. You can have a pharmacodynamic study. Sometimes you can have in vitro BE studies, or a clinical endpoint study as well may be appropriate in certain circumstances. So again, to highlight the point, while a pharmacokinetic study may be appropriate for product A, a pharmacodynamic study may be appropriate for product B. And that stems back to that regulation I talked about, the 3.20.24a, which says that the most accurate, reproducible, and sensitive approach must be used to demonstrate bioequivalence. So, as I've outlined so far, there are a number of different types of studies that can be used. So, how will we know what's on the agency's mind in terms of what we think the most appropriate path is? And for that, we have published product-specific guidances. And these guidances, as I mentioned earlier, contain, among other things, BE study recommendations for specific products. The guidances are publicly available on our website, and they're published in an incremental manner, approximately every 3 months or so. And again, these guidances will recommend in vivo studies for certain products, or maybe in vitro studies, or a combination of the two, depending, again, on what is the most accurate, sensitive, and reproducible approach to demonstrate BE. And in some cases, a waiver of the bioequivalence study requirement may be recommended. So there are a number of different considerations when we think about what the appropriate bioequivalence approach is going to be for a given product. And here is just a few thoughts. One would be the mechanism of drug release and delivery, the intended site of action, formulation design and composition, the ability to measure the drug systemically or at the site of action, and different, what is the current technology in terms of the available in vivo and in vitro tests. So there are a number of different considerations there. I think one of the most fundamental considerations really is the site of action. So, considering that the site of action is an important concept, I think that would sort of explain why a pharmacokinetic study is typically the best BE method to demonstrate bioequivalence for a solid oral dosage form. So let's think about why this would be the case. So the vast majority of this type of dosage form, the site of action is systemic circulation. So if we want to compare the rate and extent of the availability of the active ingredient from the test and reference products at the site of action, we'd want to measure the active in the systemic circulation. And so for this reason, it would then follow that a pharmacokinetic study would be the most sensitive, accurate, reproducible approach. However, this doesn't necessarily hold true for all the dosage form, or all the solid oral dosage forms. And here, let's take the case of vancomycin hydrochloride capsules. So what is it indicated for? It's indicated for the treatment of diarrhea and enterocolitis. And the mechanism of action of this product is actually on the different microorganisms that cause or that lead to the diarrhea. And so when we consider that, the site of action is actually not systemic, but is local. So getting into a little bit more of the detail here, the capsules, after oral administration, release drug in the stomach. the upper GI tract. The drug is completely solubilized in GI fluids. Then the solubilized drug is transported in the GI fluid to the site of action in the lower GI tract, which is again on the microorganism. And the other thing to consider is that vancomycin is poorly absorbed after oral administration. So when we take these concepts together, particularly the local site of action and then the poor absorption, one would think that a PK study is not an effective way to assess BE in this example. So in fact, then, for vancomycin hydrochloride capsules, the product-specific guidance recommends, first of all, that there's Q1, Q2 sameness, meaning qualitatively and quantitative sameness between test and reference products. And that is to ensure that there is no effect of excipients on the transport of drug to the site of action when comparing the test to reference products. And then the second aspect is that the guidance recommends similar test and reference product dissolution at all physiologically relevant pHs. And that's to ensure, again, that the rate and extent of availability of the vancomycin in the lower GI tract is comparable between test and reference product. So it's important to consider that while in many cases we recommend in vivo BE studies or in vitro BE studies, in many cases these BE studies are not necessary. And a number of different scenarios where it may not be necessary is listed under 21 CFR 320.22. And some of these examples here where a waiver might be a possibility would be oral solutions, parenteral solutions, ophthalmic solutions, otic solutions, and so on. And really what this is saying is that in cases where we would consider the bioequivalence to be self-evident, you might consider a waiver. And that would be the case for a solution where, in theory, you would at least have a minimal effective formulation effect on bioavailability. And then the other paradigm that I'd like to highlight here where we consider waivers would be in cases where we have a product strength that was used in a bioequivalence study, and then the applicant will then either propose a lower strength of that same product line or slightly reformulated product. In that case, we can sort of say, well, the formulation that was tested in the bio study is not that much different compared to this newly proposed formulation, so we can waive the bioequivalence requirement for the lower strength or the reformulated product. So those were waivers that I covered on the previous page are as I said, based on the idea of either self-evidence of bioequivalence or just reformulated products or products with slight formulation differences to the formulation used in a bio study. This paradigm I'm going to talk about now is slightly different. This is the Biopharmaceutics Classification System waiver system. And this is based on the intrinsic physical characteristics of the API and also the dissolution characteristics of the dosage form. So there are three foundations to the waiver. There's permeability, solubility, and dissolution. And the concept is that the agency can consider a waiver of the BE study requirement if the applicant can demonstrate that the drug substance is either a BCS class 1 or class 3 drug substance, meaning where BCS class 1 is high solubility, high permeability, and BCS class 3 is high solubility, low permeability. So why can waivers be based on solubility, permeability, and dissolution? So really, both three factors determine the absorption of a drug from the formulation, right? You have the dosage form dissolving, and then you have the API needs to solubilize in the GI tract, and then it needs to permeate into the systemic circulation. So the basic concept then is that if a drug substance has high solubility, it's unlikely that formulation differences between test and reference product would affect the rate and extent of absorption of the API, provided that, one, the in vivo dissolution of both formulations is rapid or very rapid with respect to gastric emptying time, and two, that there are no excipients that would affect absorption. So while the concept is relatively straightforward, not all products are eligible for this particular approach. So what products are eligible? So it's immediate release solid oral dosage forms with the BCS class 1 drug substance that exhibit rapid dissolution, or the BCS class 3 drug substance that exhibit very rapid dissolution. And again, in both cases, there should be no concerns regarding excipient effects on absorption. Products that are not eligible are modified release drug products, narrow therapeutic range drug products, and dosage forms intended for absorption in the oral cavity, for example, sublingual tablets. So the agency has published a guidance for industry. Right, so the agency has published a guidance for industry, waiver of the in vivo bioequivalence and bioequivalence studies for immediate release solid oral dosage forms based on a biopharmaceutics classification system, or I'll call it BCS for short. And it lays out the fundamental recommendations that we have when an applicant is applying for this type of waiver. So I'm going to sort of walk through the highlights of the guidance and hit upon some of our general thoughts on what's needed to demonstrate the solubility characteristics of the API, permeability, and also dissolution of the dosage form. So for high solubility, which needs to be demonstrated for both a BCS class 1 based waiver and a BCS class 3 based waiver, the high solubility is established when the highest strength is soluble in 250 milliliters or less of aqueous media within the pH range of 1 to 6.8. And I think it's important to note that if the single highest dose is higher than the highest strength, then additional information may be necessary to demonstrate high solubility of the API. Also important to note that we recommend that the solubility be measured under equilibrium conditions using a shake flask method. And then moving on to high permeability, which only needs to be demonstrated for BCS class 1 containing drug products. There are a number of different approaches that can be used, mass balance studies and different intestinal permeability methods. Among those that is used quite a bit is the KGO 2-cell model. And it's important to note that when using cell culture systems to demonstrate high permeability, that the expression of efflux transporters is measured, because we don't want to be in a situation where we have a false reading of high permeability. So continuing then with the high permeability, for cell culture methods, the method must be validated using a number of model compounds. And critically, we recommend that a high and low permeability standard be used in the pivotal study. The idea being that then we have nice markers there to actually demonstrate that your compound of interest has high permeability. Moving on then to the dissolution aspect of it, the recommendations are slightly different depending on whether you have a class 1 or a class 3 product. For class 1, we recommend that rapid dissolution is demonstrated, meaning a mean of 85% in 30 minutes. Whereas for BCS class 3, we recommend very rapid dissolution, meaning a mean of 85% in 15 minutes. And the test and reference product dissolution profile should be similar. It's important to note that in the guidance, we have some specific recommendations on the conditions to be used in the dissolution study, and deviations from those conditions should be justified in your submission. And then finally, as I mentioned in this part of the talk, excipient effects are a concern when we consider the eligibility of a product for a BCS-based waiver. And the recommendations are slightly different depending on whether we're looking at a BCS class 1 drug substance or BCS class 3 drug substance. For BCS Class 1, we recommend that the product does not contain any excipients in amounts that will affect the rate or extent of absorption. So a key culprit here may be, for example, sugar alcohols, something we want to look at there. BCS Class 3, we're more conservative in our recommendation. We recommend that the test product formulation is qualitatively the same and quantitatively very similar to that of the RLD product. So some general tips then on BCS waiver studies, and some I've touched upon before, but for solubility studies, we want to make sure we have equilibrium conditions. Saturated solutions should be used. We recommend that the pH be measured at the beginning of your solubility study and at the end. We also recommend the shake flask method be used, and no surfactants as well. For permeability studies, again, we would recommend that internal standards are not efflux transporter inhibitors. And another key thing to consider is that citing literature data or labeling from a non-US product to justify, for example, the permeability or the high permeability of your drug substance is usually unacceptable. So to this point, we've covered some of the basic regulatory and scientific concepts behind demonstrating bioequivalence, and we've described some scenarios in which the waivers of the in vivo BE study requirement may be pursued. To close this presentation, I'd like to take a few minutes to provide some general thoughts from the Office of Bioequivalence and our assessors that we hope will help you submit complete and high-quality bioequivalence submissions in your ANDAs. So I think my main message in the next few slides is that if we can use all the tools that are provided by the agency to industry, that will, I think, smooth out the assessment process on our end. And I think really in this regard, as I alluded to before, the main and most prominent and obvious resource that we have, certainly from a bioequivalence perspective, are these product-specific guidances. So I think we would highly recommend that you keep abreast of new and revised PSGs that are published on our website. And the links for these websites and some of these tools I'm about to mention here, by the way, can be found in the references slide of the slide deck. You can also sign up for listservs that provide alerts on new and revised PSGs that have posted. It's important to note that while the term is product-specific guidances, but some of our product-specific guidances actually have some helpful considerations for special bioequivalence topics. So for example, there is one product-specific guidance that has a description of the considerations behind demonstration of bioequivalence for highly variable drug products. There's another product-specific guidance that talks about statistical and study design considerations for narrow therapeutic index drug products. There's also guidance that talks about population bioequivalence analysis. And again, I've provided those examples in the reference slide. So we have these product-specific guidances, but we also have some more general guidances, and they're found in the biopharmaceutics section. And again, the link is in the reference slide. But I think two key guidances I would recommend would be to look at. One would be the bioanalytical method validation guidance. Again, and that guidance has a lot of useful information on some of the recommendations regarding bioanalytical method validation and measurement of samples that are made of biological matrices. And second then is our bioequivalence studies with pharmacokinetic endpoints for drugs submitted under an abbreviated new drug application guidance. And that has a lot of important information regarding recommendations on study design and pharmacokinetic parameters to measure, among other things. So while we do our best on the agency side to provide these product-specific guidances and general guidances to address situations that come up during the development and assessment of bioequivalence studies, obviously, there are going to be questions that might fall in the gaps and may not fit in, or may not be addressed in any of these other guidances. So one recommendation would be to look at product-specific guidances for similar products. The other recommendation is to contact us, and there are ways to do that. I think, as alluded to in a previous presentation, we have the ability, or you have the ability to ask questions via controlled correspondence, and there's a draft guidance that discusses the process behind that. And then also for complex products, meetings may be held as well. And there are a number of different types of meetings. There are product development meetings, pre-submission meetings, and mid-cycle review meetings. And again, the process and logistics of that are described in the formal meetings between FDA and ANDA applicants of complex products under GDUFA. A critical point, I think, to mention is that, in many cases, these contract research organizations are hired to conduct these studies on behalf of the applicant. And the bioequivalence outcome is obviously only as good as the quality of the data. And so therefore, we would highly recommend that you hire a very good, reputable CRO. Another thing that we would like to emphasize is regarding retention samples. And we want to note that per regulation, 21 CFR 320.63, retention samples are needed for bioequivalence studies. We have a guidance for industry out handling retention of BA and BE testing samples, which further elucidates that this requirement applies to both in vitro and in vivo BE studies. Then moving away from the science a bit more to the content, I think it's important to note that even if the submission has rock solid data and so on, but if the data are difficult to find, then it takes our assessors more time to find the data and to analyze the data, and that obviously slows down the process. So I think it's in everyone's best interest to provide the most clearly well-written study reports that they can, provide all data needed for analysis, complete summary tables, and so on. And again, we have a guidance for that talks about the content and format of ANDAs. Also critical to note that complete site information, including street address, should be provided for all CROs who conduct BE studies. That would be in vitro and in vivo BE studies, and studies in support of BCS waiver requests. We also have, with respect to good ANDA submission practice, there's also guidance here that I've provided on this slide. So those were some more general observations that we have. Also, I'd like to relate to you some concerns on a more granular level that have been noted by our assessors that we'd like to share with you. For example, in the realm of statistical analysis of studies, an incorrect statistical model was used, or there was no pre-specified statistical analysis plan. Some other concerns noted by our assessors as far as in vivo BE studies go are missing long-term storage stability data, inadequate dilution integrity data, either missing SOPs or SOPs that lack specific objective criteria, and so on. And also in terms of study design, sometimes we have issues with inadequate sampling time, for example. We'll have studies with a lot of first point Cmaxes and so on, which leads to concern about the accuracy of the Cmax measurement. In this regard, it's important to note that for applicants whose studies submitted after December 17, 2016, the data must be submitted in formats supported by FDA and listed in the FDA Data Standards Catalog. And you can visit this website for more information on that. And then as far as in vitro BE studies go, again, concerns on statistical analysis and also analytical method as well. So then in closing thoughts, I've sort of walked through, first of all, some of the regulations that govern bioequivalence, then moved towards some of the different types of bioequivalence studies that one may reviews to demonstrate BE, and then finally moved into waivers, and then some general thoughts from our office and some more granular concerns from our assessors on BE portion of these submissions. So just to close, I'd like to say, I think we all have the same goal in terms of providing high-quality, affordable generic drugs to the American public. And so we hope that this presentation has sort of helped shine some light on a path forward where we can achieve that